Hello, and welcome to Chem 4411 Biological Chemistry Laboratory 1. I am Professor Linda Columbus, and I will be your instructor for this semester. Before we start, I wanted to tell you a little bit about myself. I received my BA in chemistry at Smith College in 1996, and then went on to graduate school and graduated in 2001 with a PhD in biochemistry and molecular biology at the University of California, Los Angeles. My PhD advisor was Wayne Hubble, and I worked on electron paramagnetic resonance. My work focused on characterizing protein dynamics using a method called site-directed spin labeling. I then went on to do a postdoc at the Scripps Research Institute under Kurt Wuthrich, where I focused on biomolecular NMR of membrane proteins. In 2007, I began my assistant professorship here in the Department of Chemistry. What many of you may not know is that most of my time is dedicated to research, and I raise money for my research through grants, and I have graduate students in my lab that conduct experiments to answer the questions that have been proposed by my grants. The focus of my lab is membrane protein structure, function, and dynamics. We use many biophysical methods: nuclear magnetic resonance, electron paramagnetic resonance, small angle X-ray scattering, X-ray crystallography, and many more to understand what membrane proteins look like, their architecture, how they move to perform their functions, and what their functions are. Specifically, my lab is focused on membrane proteins that facilitate and mediate bacterial pathogens to infect human hosts. We are interested in bacterial pathogens because they have many functions that the pharmaceutical industry would find very attractive for drug delivery mechanisms. For instance, there are bacterial pathogens that cross the blood-brain barrier, and most bacterial pathogens are tissue-specific. Some induce phagocytosis in non-phagocytic cells, and others avoid lysosomal degradation. All of these are functions that have yet to be gained in drug delivery systems, and all of which would be very useful in effective therapeutics. Now that I've introduced myself, let me introduce the other instructors involved in this course. Dr. Price is a laboratory coordinator for the course. She has a PhD in microbiology, which she received here at the University of Virginia working with Robert Cadner. She not only coordinates the physical aspects of the laboratory, but she's intimately involved in designing the research aspects as well. She, along with Professor Mura and myself, have created, developed, and raised funds to continue the development and design of this research-based course. Your teaching assistants for the course are all graduate students in the Department of Chemistry. They are Ryan Oliver, Chris Lee, Jen Patterson, Tomek Kobzinski, Jennifer Martin, Arthur Sikora. You will get to know one of these very well as your permanent TA, but also you will meet many of the others during office hours in which the TAs rotate. I want to establish a few ground rules before we start the semester, and in particular emphasize that my expectations are high. You are UVA undergraduate students in your fourth year that have decided on a specialty in biochemistry. But I also want to emphasize that although these are high, my expectations are not unreasonable, and I am here for discussion if you ever think they are. With that said, I'd also like to emphasize a few points that I find incredibly important. The first is that I do not tolerate any disrespectful behavior. I expect that all of you treat each other, your TAs, and me with absolute respect. You also need to take responsibility for your behavior and actions. Also, I expect that you are a biochemistry major and you've chosen this major because you find the topic incredibly interesting and that you want to be in this course. So because you are very interested in this course, that should drive your self-motivation to excel and perform well in this course. You should also be willing to educate yourself. That is a big part of this course in which we're trying to teach you to be self-learners. This course is different than many that you've taken, and we're really trying to switch from a passive learning process to an active learning process. And I thought it'd be important to let you know why we're choosing to change the laboratory instruction curriculum. So first, some statistics. Why should we change how we teach science to undergraduates? In the past 10 years, it's become clear that the United States is dwindling in their performance worldwide in terms of science and technology advancement. Here are a few statistics that outline that fall in our status worldwide. The U.S. is ranked 48th in the quality of math and science education as reported by the World Economic Forum. In 2009, 51% of U.S. patents were filed by non-U.S. companies. The United States ranks 27th among developed nations in the proportion of college students receiving undergraduate degrees in science or engineering. 93% of United States public school students in 5th through 8th grade are taught the physical sciences by a teacher without a degree or certificate in the physical sciences. These are only a few numbers, but there are many reports, in which I'll outline in the next slide a few, that point to these numbers and expect that educators such as myself respond to these numbers in terms of education curriculum. How should we change the science education curriculum? I will summarize two reports that are relevant to this course that provided guidance for educators to adapt their courses to include recommendations by the National Science Foundation, the Howard Hughes Medical Institute, and many other science foundations. The first was a report in 2006. They suggested 9 urgent action items, and 2 are relevant to this course. The first is that we should be giving undergraduates the opportunity for personal experience with inquiry-based learning that brings you, the students, to deeper understanding of the nature of science, or in this case, biochemistry. The second is to be adventurous in exploring different curricula to strengthen learning in science, technology, engineering, and mathematics STEM fields, and piloting new ideas, tools, and approaches to developing curriculum, such as what we're doing in this course. There's another study and report published by the Howard Hughes Medical Institute in 2004 that, if I were to summarize it with one quote, is to teach science the way you do science. So we should be engaging you in discussion and exploration rather than teaching you facts. And that is really what is the essence of this course. This course is designed with the first semester to be more inquiry-based and the second semester to be research-based. In the first semester, we train you in the most common methods of biochemistry that you will use next semester. Each laboratory is not independent and they are related. For each week, you should be thinking about how the material can be assembled together and why that particular task is important to understanding the function of your POI. Your goal is to characterize the function of your protein of interest experimentally and computationally. Again, with each week, you should be asking, how and why is this related to my investigation of my protein of interest? If you don't know how it is related, you should ask your teaching assistant, Dr. Price, or myself. A few course housekeeping points. In addition to what's in the syllabus and your manual, you will also have mini lectures such as this one and demonstration videos to watch before lecture or laboratory. This material is highly relevant to lecture, laboratory protocols, pre-lab quizzes, and the final. Each week we will post them on the website for your accessibility, and we'll let you know which ones are important to laboratory and which ones are important for lecture. Please read the syllabus and watch the syllabus video and email me any questions you may have. All of our emails can be found in the UVA directory. Next week's lab is split into 1A and 1B. This split happens in a few places in the syllabus, so be sure to check both pre-labs and the due dates for those pre-labs. Before lecture on Friday, look up the general reaction that is catalyzed by your protein of interest. Also look up terms and refresh your memory of terms that are related to enzyme catalysis. You need to be prepared with the language of biochemistry in order to perform helpful literature searches related to your protein of interest. Please bring your laptops for an interactive lecture on literature searches that will be given by Beth Blinton Kent, our UVA chemistry librarian. To close this mini lecture, I would like to emphasize why we are focusing on literature searches to such an extent. From now until May, you'll be using the plethora of research that has been published to help you design your experiments, to educate yourselves, and to compare your results that you get in lab to those that are in literature. Every lab report you submit and paper that you write will require citations to support your ideas and add validity to the data that you acquire. Therefore, the lecture that the librarian will give on Friday is incredibly important, and you should be thinking about how to apply it to your overall goal. And remember, your overall goal is to characterize the function of your protein of interest both experimentally and computationally. I very much look forward to this semester, and I will see you on Friday.